What do you think that makes PBPK suitable for the evaluation of PROTAC pharmacokinetics?
Well, as beyond rule of 5 compounds, PROTAC have a complex ADME behavior. For example, absorption and distribution into tissue are quite difficult to predict. Eventually, their pharmacological effect depends not only on systemic exposure, but on whether they reach the right tissue and accumulate at the right levels inside cells. So PBPK, physiologically based pharmacokinetic modeling, when applied thoughtfully, is a powerful framework to link absorption, tissue exposure, and ultimately also pharmacological response, making it highly valuable to understand and predict product pharmacokinetics. In fact, PBPK can integrate physicochemical, ADME properties, and generate mechanistic pictures of product disposition.
What kind of targets would benefit more from an intervention with PROTACs, and how do you think that PKPD can help to inform that question?
Yes, selection of target is very important when it comes to PROTAC. We have to first remember that target such as protein are not a static thing. It is constantly being turned over. That means it's being constantly produced and degraded. So for PROTAC to work well, you want a protein that turns over fairly slowly, preferably over several days. That means that once the PROTAC degraded the protein, it will take the protein several days before it can come back. And that is where the power of PROTAC lies. If a protein comes back very quickly, say within a few minutes, in the other extreme, then obviously the power of the PROTAC becomes much, much less effective. So yes, it's very important that we pick the right target. Now, one thing to keep in mind is that the protein turnover is typically much faster in rodents than in humans. So if you had tested your PROTAC in an in vivo animal model, you may not get as effective result as you would like to see in human. That doesn't mean your PROTAC is not working. It just means that you're testing that in a species that protein turnover is faster than where the species that you ultimately want the PROTAC to work in. And this is why it's so important to have PBPK and PD model to come together to help us make these decisions.
What kind of methods can be used to translate protein degradation data typically produced in vitro or in animal studies to predict the human PD?
This is actually a continuation of the previous question, where I already started discussing the importance of translating the different species difference in protein turnover to predict human effectiveness. And in fact, if you have tested the preclinical in vitro system rather than in animal, that turnover rate may also be different, right? The protein turnover rate in the cell, in the test tube may also be different from that in human in vivo. And so those all had to be translated. But that's only one of the many things that need to be translated. How well can the drug get into the target cell may be quite different between animals and human and in vitro system. Also, that the expression level of the E3 degradation enzyme is well known to have huge species differences, as well as the binding affinity of the product to each one of its binding partner can also be different. Now, we have ways to test or quantify the difference in each one of these elements. However, to tie them together to say how they work together to define the effectiveness in human, now that's where we require the PBPK/PD modeling to help us, bridge all this information into one to estimate the human effectiveness. And this is why in our webinar, we're going to be emphasizing on the importance of modeling to help us get there.